Okay, so today we're going to talk a bit more about actually the practice of cloning, how we'd actually do it in the lab. Sorry, these lights still, obviously, if I don't say anything no one else has noticed in any other lecture that they've been in. Anyway, so sorry, but we have to have the lights all on or all off, I guess. Okay, so these are the things that we're going to cover today. So we're going to look at how different forms of plasmid DNA run on an agarose gel. So this will be relevant because you'll be doing that in your practical. We're going to look at how different types of enzymes are used in molecular biology. So we started already talking about the fact that, you know, some enzymes like the RNA polymerase, we know how it works, or a reverse transcriptase that we use in this reverse transcription PCR that you did in your pracs this week. We know how that works, and therefore we can use it as a tool. And so we'll have a look at a few more examples of that, of different enzymes that are used normally in the biology of different organisms, and they've been taken and now used as tools in molecular biology, in cloning, etc. Okay, so we're going to have a look at how you would use restriction endonucleases and how you would use specific sites to clone a DNA fragment, like your gene, into a vector. We're going to have a look at what it means by cloning in frame and why that's important. So this would be the reading frame. We've talked a little bit about this already. And we're going to have a bit of a brief discussion on how PCR works. So you will have already done some PCR, and next time you go to your practicals, you will get to run the PCRs that you've done in your pracs and see whether they have worked. And then we'll kind of finish on the polymerase chain reaction and have a few examples. So to give you an idea on the types of examples you would need to look at when you do your assignment that's about the PCR, polymerase chain reaction. Okay, so our plasmid DNA, although it's always depicted as a circle, so a double-stranded circle, it actually exists in different forms, both in the tube and in the cell. If you transform this plasmid into bacteria, into Escherichia coli, so the linear would be the form of the plasmid that we talk about when we talk about the linear plasmid. So the first thing, so it's just the linear double helix, would be if you cut your plasmid with a restriction enzyme and then you linearize it. Okay, so you give it one cut, and therefore it's now a linear piece of duplex DNA. Nicked DNA is essentially when it's the typical way that we depict a plasmid, so in the circular form. So we refer to this as it being relaxed. So it's circular and it doesn't have lots of coils. The next one is essentially very often how you find plasmid DNA, especially in bacterial cells, is that it's all coiled up and again very compact. Okay, so the same idea is even plasmid DNA, when it's in its native environment or it's in its host, in a bacterium, it's all coiled up very, very compact. And we refer to that as supercoiled DNA or supercoiled plasmid DNA. And essentially you have covalently closed circular DNA, and the supercoiling gets done by another enzyme. Again, we have lots of different enzymes that do lots of things to the DNA. It's called a topoisomerase, and it essentially coils up the DNA and allows it to be more compact. So these are the three forms that plasmid DNA can exist in, and if you look at them under an electron micrograph, you'd actually see that. And so this electron micrograph is essentially going from the left, where there's a circular plasmid, so a nicked plasmid, so it's a relaxed circular plasmid, all the way to something that is coiled up, coiled up a lot, and therefore starting to become more compact. Okay, and so essentially these forms of plasmid DNA, although you won't see them when you run a DNA agarose gel, you actually see the DNA running migrating at a different rate when you run these different forms of plasmid DNA on an agarose gel. So we'll have a bit of a look at what that looks like, because you'll be looking at agarose gels quite a bit in your practicals, so it's useful to know what it is that you're going to be looking at. Okay, so this is a typical agarose gel. So this is the gel matrix that we use to run DNA on, and it's essentially if you load your samples at the top of the gel, and the samples run from the top to the bottom of the gel, or it's flat, but it's essentially running from the top of the gel to the bottom of the gel after you apply electricity. Okay, and DNA being negatively charged, you start off at the black end or at the negatively charged end, and the DNA will run to the positive, so to the red end of the gel tank. And that's kind of important, or you will end up running your DNA off your agarose gel and not into your agarose gel. So DNA gel tanks are usually set up to not allow you to put the lids on the wrong way, to make sure that you don't plug in the gel the wrong way, but it's useful to make sure that you're running it properly. Okay, so essentially these three are the three forms of DNA that we talked about. So again, linear, so it'll be just the plasmid that we've cut open with a restriction enzyme or something like that, well, only a restriction enzyme. Relaxed, so it's the DNA that has one nick in it, which means that all of this coiling that occurs in the supercoiled has been released, and you just end up with your circular plasmid and then the supercoiled. So we've got the nicked circle runs at a particular, so the bigger things run at the top of the agarose gel, and smaller things or smaller looking things run towards the bottom of the gel. So interestingly, although these three things of plasmid DNA are the same plasmid, they migrate at different rates in the same DNA agarose gel mix. And that's essentially because of what resistance the DNA encounters as it moves through the matrix. And that really has an importance on what the DNA looks like. So the DNA that is supercoiled, although these three are the same bits of plasmid DNA, the supercoiled would run much quicker because it's a tighter little molecule. Okay, so it's the same size of base pairs, but because it's much more compact, and the agarose is essentially a matrix, a compact thing ends up looking like it runs quicker through an agarose gel than the other two. So if you cut the plasmid DNA, it runs at its true size, essentially, and so once you restriction enzyme digest your plasmid with a restriction enzyme, and you run a molecular weight marker, which you will do when you run your gels as well, the size of the plasmid, when you linearize it, it's the reflection of the true size of the plasmid. So say this plasmid is 4,000 base pairs or 4 kb, it would run exactly where the molecular weight marker is, which is just a ladder of different size bands. You would be able to line it up with something that was 4 kb, so the size of the real size of the plasmid. So the nicked circles runs, gets retained, even though, again, these three are the same bits of plasmid DNA, because it's a nicked circle, so it's a bigger object running through a physical matrix. It just gets retained in the gel more than the very compact supercoiled form or the linear long bit of DNA. And so it looks like it runs slower on a gel, and it looks bigger, but they're in fact three of the same plasmid, just different forms. Okay, so because a gel is just a matrix, just a physical matrix with kind of little pores through it, and smaller things go through it quicker, or smaller appearing things will go through it quicker, and larger appearing things get retained or get stopped by the matrix. Okay, so we also use different percentage of this agarose gel matrix depending on the size of the DNA fragments we want to actually see on the gel. And So if you're looking for fragments that are small, then you would use a larger, an agarose gel that has a greater percentage of the agarose. And you can see this from here. If you the gels here, there's 0.7% of agarose, so that's quite a low percentage of agarose to use. Or 1%, which is what you'll be using in your practicals. So 1% agarose is kind of the standard, unless you're looking at very small things or very large things. And then if you're looking at very small fragments, you would increase the amount of agarose in your agarose gel to something like 1.5 or even 2%. And so you can see clearly here that if you're looking for large bands, so again, this is the top of the gel, this is the bottom of the gel. The top of the gel has the big things, and then as you go down the gel, you have smaller and smaller things. And you can see in different percentage gel, we're running exactly the same thing on here. And this is a molecular weight marker that you'll also run. But you can see that they get dispersed quite differently depending on the percentage. So a lower percentage gel is good for larger fragments, to resolve larger fragments. And you can see these DNA fragments here. There, you can actually see a ladder of large fragments. While the smaller ones, you're starting to lose resolution. You can't see very, very clear bands anymore. And as you increase the percentage, you can see that the large DNA fragments start to bunch together at the top of the gel. We can't really understand what they are, but the smaller fragments start to separate out much clearer. And then if you go to even a larger high percentage gel, if you're really interested in very small things, so if you're looking to see whether the gene that you've cloned into your vector, it's a very small one, say, I don't know, 100 base pairs or 70 base pairs, you'd want to be using a higher percentage agarose gel. It doesn't resolve the big things, but you can see much better resolution of the smaller fragments. And so obviously you have to know the size of the fragments that you're going to run on your agarose gel to be able to determine what the best resolution range is for the fragments you're looking at. So we're running a 1% gel because we will have our plasmid running on the same gel, as well as the cut plasmid with all these bands that we're going to check whether our insert has been inserted into our plasmid by digesting it with this BTS1 enzyme. So we'll see a whole ladder of different sized bands. So a 1% gel is a good place to start, because you can see kind of both ends of the scale. Okay, so it's important to know what size fragments you're working with to decide what percent agarose gel you're going to use in your DNA gels. Okay, so the enzyme. So we're going to talk a little bit about the fact that all of these different enzymes that we use in the molecular biology lab as a tool are from different organisms, and they perform, whatever specific function. These enzymes perform specific functions in the organisms they come from. So this is a table that just goes through some of the enzymes that we use when we do cloning. So recombinant DNA technology is everything that you use when you're cloning. So there's lots of different things that you need to do to prepare your genes, or some depending on what you're cloning and how you're cloning it, and for what reason you're cloning it. You need to process the thing that you want to add into your vector, or the gene that you want to add into your vector. So you can see we're going to talk about the first one, which are the restriction endonucleases. These are the ones that cleave the DNA at a specific base sequence, and this is essentially how we still routinely do cloning in the lab, and we're going to talk about this in the next slides. So we need to have a DNA ligase when we actually perform when we insert our gene into our plasmid. You need to make a phosphodiester bond between pretty much the end of the plasmid and the start of your gene, and to make a bond in between those, or else you're not going to end up with a circular plasmid that won't then be able to be replicated in bacteria. And there's lots of different, and so reverse transcriptase would be the enzyme that we used in our practicals this week, where we're making a DNA copy of the RNA of our expressed TP63 gene. The DNA polymerase you've also already used. This is what you use when you do the polymerase chain reaction. So this is the essentially the enzyme that's going to duplicate the DNA that you want to make. So a lot of your TP63 gene in the PCR tube, you need a DNA polymerase to make a new DNA strand. And there are lots of other enzymes that we also use for different reasons in molecular biology, and we're going to have just a look at these two, this exonuclease and this alkaline phosphatase, because we're going to talk more about these as the lecture goes on, and actually even towards the end of the lectures, especially the exonuclease activity. So the alkaline phosphatase is an enzyme, obviously, ACE phosphatase, and as the name suggests, it actually removes 5' phosphates. So if you have a DNA strand, you'll always have a terminal 5' phosphate. Okay, so that's the end of your DNA strand, and that's where, remember, where the phosphodiester bond is formed between one DNA base and the next one is through that phosphate, through that phosphate group. So anyway, so all DNA strands will have this terminus, that is a 5' phosphate, and we use the action of this enzyme when we perform ligations. So it's, we're going to talk again about the type of ligations you're doing in your practical. It's slightly different. It's this TA cloning. But when we perform kind of normal ligations with restriction enzymes, we actually use the alkaline phosphatase as one step before we do the ligation. And we'll talk about that in the next few slides. The other activity I wanted to show you, again, enzyme. It's an exonuclease. It means it chews away from the ends. And if you, we'll talk about this in our DNA repair lectures, because many of the DNA polymerases that we use in the polymerase chain reaction have exonuclease activity. So what that means is that, say they've put in so this is the correct DNA base. So say they've put in instead of putting in a G here, they've put in an A by mistake. So DNA polymerases are don't make many mistakes, and the reason why they don't make many mistakes is because they read what base they've just put on the DNA. So if they find that they've put on the wrong base, so they've put on an A here, the exonuclease domain in that whole big enzyme will read and see that it's put on the incorrect base, and will break a phosphodiester bond, okay, and remove that DNA base that is incorrectly put in there. So we'll revisit this, but again, the idea of having the function that we use in the molecular biology lab is essentially the function of different enzymes, and we exploit those as a tool. Okay, so we're going to talk now a bit about restriction endonucleases, because, as I said, it's still the most kind of routine way to do cloning. So if we remember from last week, we talked about these all plasmids or vectors have this multiple cloning site with all of these different restriction endonucleases that you can choose to stick in your gene of interest into that plasmid. So restriction endonucleases they've been around for a while, or we know their existence for a while, and the reason why they are apparent is that in bacteria they essentially prevent invasion of foreign DNA. So, for example, viruses or etc., or phages, these types of things. And so what they do is that they cut up foreign DNA. So any DNA that's not part of the bacterium is cut up by these restriction endonucleases, and they have obviously a lot of them, and therefore, you know, any foreign DNA will end up being cut up, you know, many times by all these different restriction endonucleases that different bacteria have. So they're termed endonucleases because endo is inside, so they cut a DNA sequence from within, so they don't need an end, and that's why they're called endonucleases. If you have an enzyme like a exonuclease that cuts from the end, it's called an exonuclease, so from the end, essentially. you'll see that, and you'll already have seen